good morning, good afternoon, and good evening to all of our customers and partners and guests who have joined us from across the globe. My name is Rahul Madhavan, and I serve as Point Cross's Life Sciences Vice President for Strategic Programs based in Washington, D.C. As we shift to the next slide, I wanted to frame today's conversation and your understanding of the Xbiom platform as a recommendation engine for data mapping to various exchange standards. Our objective is to substantially reduce the programming burden and to enable greater QA and oversight. Our goal in releasing Xbiom is to get users to complete their analysis with speed and fidelity. This involves ingesting unstructured raw data and metadata from CTMS, EDC, biomarker data to a longitudinally integrated view of any trial at any time, whether clinical or non-clinical. And because of the continuous cycle data ingestion and mapping to an invariant universal data model, which you're going to hear more about today, teams across sponsors have ready access not only to individual or highly stratified cohorts of patient profiles, but a curated data set readily mapped to an exchangeable data set. So let me now turn to today's presenter, who assuredly many of you on today's call already know, Sunil Gupta. And if I can ask Sunil to just go to the next slide. Sunil brings over 25 years of experience in the pharma industry and has authored 5 SaaS books. He's a global SaaS developer. He's a CDISC SME. He's a corporate trainer and a consultant. He also teaches practical R for SaaS programmers and a CDISC class at the University of California, San Diego. And we've been so excited to partner with Sunil, who's helped us in testing and validating the Xbiom system and its standards management and governance. So before I turn it over to Sunil, a couple of housekeeping things. Today's presentation, we absolutely encourage questions which can be submitted via the Q&A function at the bottom of your Zoom toolbar. We're going to be aggregating and anonymizing all questions and then providing written responses, which are going to be sent to all of today's participants and will also be posted on our website. Please also look out for the registration link to the upcoming Fuse Wednesday webinar, which is going to be held on January 25th. And Sunil is going to delve further into leveraging the universal data model to streamline the pathways to SDTM and ADAM standards. We're also going to be live in person at the Fuse Connect conference in Orlando between March 5th through the 8th, and we really can't wait to see many of you there. So again, please ask questions throughout at any time through using the Q&A box on the bottom there of the toolbar. And Sunil, I'm going to turn it over to you. Thanks so much.
Great. Thanks, Raul. I appreciate that. And I would like to thank everybody for their time and attention. I know your time is very valuable, especially these days, especially talking on this topic, which is very passionate to me, specifically talking about metadata standards and automation. And also, I really appreciate Point Cross asking me to put together and share what I know and my experience working with their tool, Xbiom. As you may know, working in this industry for decades, we're in the process of streamlining, namely best practices in everything that we do, as far as working with the clinical data, working with the SDTM models, and so forth. And many things along the way, checking for compliance, creating the defined XML, SDTM packages, and so forth. And so as the goal to standardize becomes paramount, and by the way, I've also, as I mentioned, been on the CDISC 360 committee several years ago. As you saw from the quote, the objective is pretty tall order to automate as much as possible. I mean, leveraging metadata to the fullest. Many of you may know a little bit about metadata, but what I really admire and appreciate using a tool like Xbiom in the clients that I'm working with is that it really leverages metadata to the fullest. And so in this slide, we're talking about the different types of roles and people have their objectives, whether you're managing and a director, managing studies, a programmer, scientist, or statistician. What I found is the tool offers a lot of features. as far as managing the study, you can definitely get better managed timelines, because it takes the whole process, which we know and are very familiar with, and then standardizes it, and that helps to really automate it. So the project management takes less time and budget. As far as task programming, managing the submission process. So the concept of low code is what I'll be kind of introducing, and the facility to create less programming to create the SDTMs, atoms, and so forth. In addition to that, mapping specifications, which is really important, because I'm a big fan of documentation. Scientists, they have better access to the data. The concept of the universal data model, which we get into more detail, really facilitates the early exploratory analysis, even before atoms are created. So what I realized early on, Point Cross's, some of the earlier clients, customers were actually scientists and statisticians working with biomarker data, and they really needed to ingest, correlate, and harmonize a lot of different raw data. And of course, working with the statistician. So having predefined templates for tables, listings, and graphs. So in this package that we're going to be going over, it's really considered like a paradigm shift. So if you can imagine, of course, we're all familiar with using Google to get answers. The universal data model offers a way to really ingest, catalog, index, and link all related data, which I'm going to be showing a little more detail. It's a really paradigm shift in the way things are being done, not really linear, but then the traceability is still there. And along throughout the process, compliance is paramount. So the Xbiom uses the universal data model, which I'll show you 6-step. It's a visualization process. The design of that, where it really facilitates repeatability, usability, and then of course the SDTM generation, the step-by-step process, which, as you can imagine, mimics what's done, manually. And I consider this to be like evolution. We're originally starting off as SAS programming. Then we have introduction to the CDISC standards. We really set the true standards, meaning the model concept. And that's really where the universal data model kind of stems from. And then you have the GUI metadata mapping. And that's where the Xbiom comes in, because the metadata is only as good as it's actually being used. And the interface to the metadata becomes very important. Now we're familiar with the things that are there in the value chain. The thing that I want to focus on is the data curation, because I don't think organizations spend as much time fine-tuning and understanding the whole process. The data curation is a repetitive process to really optimize the data and metadata to ensure valuable use of the data. So that really means knowing your data, the issues with that, what are the structure, unstructured, unstructured, the metadata, of course, the components, giving us information, not just the basic metadata, but really the more what I consider to be more customized and more specific metadata, namely maybe number of observations, maybe number of patients, and so forth. So in this process of data curation is really utilized in the Xbiom and what we're going to be going over in this whole process cycle. And as I mentioned here with this type of diagram, we work with a lot of external data. Oops. We work with a lot of external data, and so all the data sources have to be indexed, cataloged, and organized. And usually, with the refresh of the data, it gets utilized further in the process. So basically, what you're doing is you're building up a system. And the key thing about this is we don't just do one study, we do multiple studies. So the studies that we do, we usually leverage the process, any code, everything that's there. And so setting up the process for one study organizations can really have a compound effect and utilize that same type of process templates there for multiple studies. So namely, setting up the structure, building it in with the documentation and creating the UDM type of model, which I'll be showing you, which in turn enables for early exploratory analysis, as well as the standardization and automation of SDTMs. Now, this is one of the process flow diagrams that you have available at the end. And what you see over here is references to two documentations. One, of course, is the CDISC 360, and another one really focuses on leveraging metadata. So when we're talking about metadata, the key things to really focus on is that you can really create metadata of most anything. And metadata is extremely valuable information that's machine readable. And so in this process flow, what you see over here is input, leveraging metadata as much as possible, leveraging, so the metadata coming in from all sources of input, not just the raw data, but also the models that are there. And then the transformation, as we know, is converting the raw data to standardized. That standardization has a lot of benefits, as we know, in the review process with the reduced review cycling time. And so there's evolution that's going over there. And as a result, what we expect to see is not just the deliverables, SDTMs, and add and so forth, but we also see the documentation, which is the defined XML, as well as compliance, making sure the compliance is there, which is the SDRG. So in this process flow, again, we're very familiar with this, working with the protocol, SAP, and then we have the raw data, creating the SDTMs, and atoms and so forth. But what I really want to draw your attention to is that there's actually metadata throughout the whole process. Many organizations may not have fully utilized the ARMS or the protocol metadata that's there, but there is actually metadata throughout the whole process, and if we fully utilize that, then we assure ourselves of a higher compliance and in a more controlled and automated type of a fashion. So this is another process flow diagram, which helps to illustrate the concept of what has been done traditionally, and then what is the future state. Because I can see this being in this industry for decades and doing things manual. I'm actually in a very good position to kind of somewhat audit what's being done in this tool, in this automation type fashion. One of the risks that you have is you feel skeptical and then feel like losing control. But in fact, what I'm seeing is there's a very structured approach. I really like the balance that's there between best of both worlds, between having the structure and the customization. So instead of doing the traditional programming activity, which we're very familiar with intense macros, batch processing, and so forth, the better approach is actually using and leveraging metadata. And what you see here in bold are where metadata is there in this whole process. And so going from the raw data, we see going from left to right, and you see from the top, you see the data with the raw code list, metadata, and then the user interface, which is the machine language productivity, really capitalizing on the metadata. So anything that was done programmatically needs to be a way, an avenue to do that within the user interface. Xbiom has that facility, whether it's joins or transposing. And one also the risk of auto mapping is you feel like you may lose control, but there's 100% user confirmation of the mapping that's being recommended. So if we go from left to right, raw data, metadata, Xbiom tool to CDISC standards, we have a standardized way that we can do this. And it's very clever the way the design of this metadata approach is. Again, it uses the hierarchy, uses the data structure that's there, the variables, the attributes of the variables, the type and everything. Then it goes down to the granular level of the control terminology, and then we'll see that in a little more detail. So once the deliverables are produced and we have the documentation is extremely important. And the nice thing about this tool is that it's all within one package. So you don't actually have to leave Xbiom in order to create a defined XML. So what you have is you have everything in sync, which is extremely important. So this is the defined XML, which we're very familiar with. It's it has the links integrated, and not only that, the specification to updates to define XML is with the system itself. And then of course you have the compliance component with this, which is the SDRG. And the also nice thing about this is it's built into the system, so you don't have to get out of Xbiom. Everything is within here, so you can actually check the compliance of any of the SDTMs along the whole process. So this just shows you again, Xbiom meaning a statistical computing environment, because it really can take and process various sources of input data. And actually, Xbiom has been used on hundreds, if not thousands of clinical studies. And one of the things as I was looking to see this feature's capability is, can it also produce the non-standard SDTM, such as the supplemental, RelRec, and FA. And actually, I was happy to see that it actually can. So instead of the linear process that we're typically familiar with and have been doing for decades, what we have is we're introducing more of a layered process. First, the concept of using the universal data model as the intermediate step to standardize all of the raw data that's coming in. Again, you can kind of think of that as similar to what the SDTM model itself. So there's not that much of a difference, but of course, SDTM model, there are various implementation guides, and so the fine-tuning changes for that need to be definitely applied. But if we take a look at the layer type of approach, then what we have is we have the raw data that's coming in, needing to be standardized. That's one of the first steps into universal data model. And then we can add additional things to it, which is listed over here, including derivations, again, which I will show you. So it's a very structured type of approach. And then what happens throughout the process is that then we're able to do some early exploratory type of analysis. So normally we would only get to this point when we get to the atoms, but in fact, with this type of approach, it actually offers us a more flexibility, again, a non-linear approach, because once you have in a standardized format with the derived variables, it's actually good to go for analysis, which is part of a nice GUI type interface. Now with this Xbiom, there's several modules, as I'll be showing you, just shows you that it has really full capability. Again, meeting the needs of the clients that the Point Cross serves. It has all the components that are needed. So it's a complete solution that's really worked well. Everything, all the components are all in here. The smart transformation is what we'll be going a little bit more into, that really enables the automation of the SDTMs. So again, universal data model, you can kind of think of this as a way to unite all of the dispersed type data. As we know in clinical trials, everything is linked so that we have a single truth. So drilling down, you can see that this is very, very similar type of structure that we would see following SDTM. So it's not that far off from that. Internally, the universal data model has a naming convention and so forth. But the key thing is to pull it all together so that everything is united. And so, as I mentioned, with this evolution, ideally using the models that are there from the CDISC standards, which define the specification structure and the specification of the SDTMs, and then of course control terminology, leveraging the metadata that's available from the raw data, having the technology to utilize machine readable information, lookup tables, and the document that, and then replication and automation for this. And that comes with several studies being utilized in order to fine-tune the system. Again, once the first study is set up with the whole process, then again, that can be leveraged for other studies. Now, again, I'm a big fan of metadata, and I put this slide together to show that many organizations and many people, many programmers may only scratch the surface of using metadata, which is towards the bottom of here, where the variable values are, or maybe files that are maybe in a folder situation. But if you really, want to use this, and as a developer, I know personally that once a process is in place, in order to prevent reinventing the wheel, it's well worth it as far as strategy and long term to correctly define the process, meaning what exactly are the inputs that are coming in, what are the functional requirements, and then take a look at the outcome. What are the outcomes that need to be produced, and in the process of what exactly is needed, what can be standardized in the form of what types of questions, maybe the variables, values, and so forth, and then creating a system so that input can be received from a user. And so Xbiom also offers that type of system. We have a best of both worlds. Input can be received by metadata as well as input in order to have some low code that's there, encompassed around a structure that really generates the output that's desired. And so again, what you have is you have a modified type of approach. You see UDM as part of this process. You see much shorter cycle time. Once again, the process. So this low programming. So and so that eliminates the need for doing a lot of testing, writing the code and documentation, because this is a system that has been used across many major pharmaceutical companies as well as CROs. And so you have a cycle that you're producing. That cycle inherently, because it's very well defined and the results are monitored very closely, that reduces the cycle time. So whenever it comes for the final version, it takes much less time because it's all incorporated within the system. Now, without going into too much detail, I do want to mention that this system does use machine learning. It's based off of many hundreds of studies. But again, conceptually, you can see what's happening is the harmonization. What you see is the cross-reference comparison at all levels between the raw data and the SDTM models and the control terminology. So with that, as you can imagine, being the developer, I could just briefly explain that there's a lot of processing, looping going on, again, cross-referencing at all levels to kind of align everything that's there. Once for a given study, the alignment is there, the system recognizes that these are the raw variables, raw data sets, raw variables, and then mapping specification to the SDTMs. And then it leverages that or remembers that because as you go through with the user confirmation, it documents that. So you can imagine various types of lookup tables. It's all really well integrated into this type of system. This again just shows you a little bit of behind the scenes of what happens with this process. So you can see that it's very comprehensive because the expectation is that it should match up wherever it's possible. Ideally, with the raw data being closer to CDISC, that increases the chances of with the auto mapping because everything would be much more in alignment. This again would be what the UDM, again, you can kind of consider this as a metadata repository. Many people are very familiar with metadata repository system. So basically what that means is that the system understands and is in very well control of all of the data that's available. And it links everything. So it knows changes, updates that are made. It seamlessly links everything together. So when you get to the interface where you're taking a look at some exploratory type of analysis, you don't have the need to link or join information. You can kind of think of Xbiom, analogous to that would be similar to maybe what SAS did a long time ago, which would be the SAS Enterprise Guide, where you didn't have to write SAS procedures. Xbiom really provides a way to standardize the submission process cycle. And so with this, what happens is, information is more readily available. Just as a reminder, this is the ARM, which if you take a look at it, provides a lot of useful information. And the advantages of having this serving as a metadata is really paramount. So what that means is the code itself can read what's in here. This serves as a specification or a blueprint, and the code reads information from here. It knows exactly what to do with that bit of information. So in the sense, the information that you put in here is very important and independent of the code. The testing will basically process and execute this. That's really the whole concept and benefit of the metadata type of approach. And the also key benefit of this, of course, the creation of the SDTMs is really to get early monitoring and resolution of data issues. And so that can be possible with the UDM type of model. So whether you're taking a look at critical data issues such as missing values, invalid dates, any of that type of stuff, zero records, or maybe study specific data issues. This enables you to have quick and early, easy access to that, which is extremely important because you can identify issues maybe at the site level. You can fine-tune maybe exploratory type analysis or various things like that. So keep in mind that many of the things that we're talking about here can be defined as metadata type information, and with the Xbiom system enables you to do that. And this just shows you a little bit of the interface that's there. So keep in mind that this is a user-friendly type interface that's designed to make it easy to pull information from various sources. So it's a matter of drag and drop, and it's designed for statisticians and scientists in mind. They're very, very familiar with the data, and they don't have to worry about the technology of linking. So whenever you use this type of interface, which I have working with statisticians to take a look at the SAP and further define cohorts, whether cohorts are based on various types of conditions, maybe single or more, or based on measurements. So you have that complete flexibility over here, and in keeping in mind that this is no programming needed for this type of interface. In addition, what you have available, again, immediately with the benefits of this type of approach, and without actually having atoms, is predefined templates. So these have been tested. They're defined by the domains. So, and this makes sense. So with experience that we have, decades of experience working with clinical trials, getting to the point of SDTMs, getting to the point of atoms, and then SAP with the TLFs. There's some standardization that's there based on the type of data that you're working with. So what Xbiom has done is what I've seen, and I'm very impressed with this, is they've leveraged that. They've seen the patterns, and so they define templates. So this really helps enable a quick jump start. So you can start off with this, and you can fine tune this. So it really enables a nice playing field for working with the data, and then you can fine tune that. As you can see a little bit here that these tables are dynamic, and so you can click on it, you can drill down, and you can really see specific issues down to the patient level. So for any type of exploratory type of analysis, whether you're taking a look at protocol compliance or safety or efficacy, and being a non-programmer, you have quick and easy access, early information, so you can see what's actually going on even before the atoms are created. And this just shows you a little bit about the auto mapping, which we'll be going into detail. As I mentioned in the earlier quote from CDISC 360, is the automated as much as 80%. And this process really helps to facilitate that. This shows you a little bit about the mechanics of what needs to be done as far as the variable mapping, whether it's direct or transformation, transpose, reducing to one file, then the control terminology, and then the 100% user approval method, and then the derivations. And the nice thing also about this is it has the best of both worlds, meaning that you have this type of structure, but then you also have places where you can insert code in order to make this happen and have that customization. And this just shows you a little bit about the mapping. that's there, going through the process from the metadata file to data file to the actual values, which we'll be getting into more detail soon. This is a 6-step process in the Xbiom, and then just the 2 features that we're showing in the demo slides are really number 1, the clinical study workflow, the data curation, which is auto mapping to the SDTMs, and then a little bit about the QC dashboard. So, but it is a very comprehensive visualization type of a tool here. Then this clinical study workflow module, what you see over here is a way to select, load the study. You can create a new study within Xbiom, and so this is where you go about doing that. The various different panels here. Definitely, there's a little bit of a learning curve, but what I found is I found that there is organization and structure. In here, you can define the CDISC specification, and you see the most recent. So I'm really appreciative that Point Cross is on top of current models that are there. So you have complete compatibility, you have traceability, you have the control terminology, you also have the validation rules. So they're very much in tune with that, because their customers have that expectations. In here, it just shows you a little bit about the importing of the data, EDC, any type of raw data. So they have a directory structure that's being utilized, and they bring that in. So there's some intelligence in there, which is really nice. And then getting into the smart transformation module, where again, it leverages the Xbiom metadata repository system, so the data that's already in there, or it can actually access external raw EDC biomarker data using the UDM type of a model. Now, the whole concept is based off of these steps, and it really, this is the heart of it. It really leverages and applies the 80-20 principle. So you can see over here, there's an initial batch process, which we have screenshots for the from the demo. Batch process defines the source input, the target, the auto generation. So again, this really mimics what's being done manually. So it makes a lot of sense. Again, there'd be internal macros embedded in this process. The dataset transformation, this is an opportunity to insert some basic code. This is what I consider to be low code. In the time of SaaS Enterprise Guide, you can kind of think of this of places where you can insert your own code. The metadata mapping is the real heart of it, where you can see the granularity down to the values. And I really like the preview where you see the raw data value as well as the standardized value. Again, before SDTMs are created, it's a very interactive type of approach. The terminology, this is where the control terminology is, derivations, and then data updates. So just jumping in, you can kind of see the interface is very user-friendly, drop-down type menu. Again, we because it's actively being utilized. So this is the interface that really defines the settings, which models you want to use, where the data is, and then the auto generate. There is straightforward process to check the box that you want for the variables. As we know, these are to be created, and it's to be done across the domains. This opportunity to write some simple code that will actually merge some raw data sets into a single data set. And that single data set will be available in the next step in the metadata mapping. And so, again, this is where I was originally kind of skeptical, saying, how can it know? How does it know if there's some joins that need to be performed? Where exactly does that happen? Well, it actually happens over here. So you have to, again, know the data and make the files available to the system. And you can do that at this step where you can join to the raw data sets available, and then that file will be available in the next step that's there. And you can see here in the metadata mapping, which is really key. On the left-hand side, you see the raw data. And I really like this type of approach, because as you know, as programmers, we have to map all of the raw data. That's a given. And that's either going to be mapped to the standardized domains or the supplemental domain. But also on the right side, where you see the target domain, we have to make sure that we have a map to the raw variables. And so here you can see several types of decisions that were made. Previous decision approved, the machine recommended based on the symbols that are there. And then the values that are being displayed on the bottom, the source and the target. Taking a deeper look at the raw data that's quickly, readily, easily available. And this is a really nice way interactively to make decisions, to review actual data values. And then, as I mentioned here on the target side, a list of all of the domains. Other domains that also are there, like the oncology domains. So the experience of Xbiom across many studies is evident as listed from this dropdown, and it makes sense. So obviously, with this approach, the user definitely needs to know how to operate the tool, and so with the recommendations that are there, and you can see the level of the target. So it helps you with not only the name of the variables, but also the label. So really facilitates that process of the mapping. In addition, it does a great job for transpose. As we know, lab data often comes in a different structure and needs to be transposed. So you can see how this works. On the left-hand side, you see the original raw data that needs to be transposed just from a click. And you can see what that looks like with the converted. And as we know, with the transpose, that version makes it readily easy to construct the LB domain. And so again, no programming, it's designed this way because it has raw data that it has expected and received like that. In addition, you can also write, again, low code. These are simple expressions. It enables you to do that very straightforward. This is the creation of the uSubject ID. And so again, the system captures all of this that you're doing with the interface. It captures it, and behind the scenes, it actually builds the mapping specification, which is really great because it really serves as automatic documentation of what's actually being done. In addition, you could also write scripts that have like this case when. So you have that flexibility, which I consider to be best of both worlds, where you can use the recommendation that's there, or you can write a simple script in order to make that happen. This shows you a little bit about the supplemental domains, that it has the facility to process and work with that. Again, it knows that because it's heavily involved in knowing the metadata of the CDISC SDTM standards, and knows what is considered non-map. So that would be putting into the supplemental domain. In addition to that, one of the things I wanted to confirm is the RelRec. As we know, this requires a little bit more knowledge, understanding. It's a very different type of domain, and it requires some special attention. So the system recognizes that, it reads that information, it links the information correctly, and you can see that over here with the results. As we see, AE and CM would be typical in the RelRec. It also has the intelligence to define what the rel type is, whether it's one or many, and you can see that very linked here. So the tool basically offers a way to view this information as you're dynamically setting and using the parameters. In addition, findings about. Findings about, again, is one of those specialized domains, and Xbiom has a facility to process that, because it recognizes there's a parent domain with that. So it knows all the variables that are there. It helps with the mapping to get to that type of structure. In addition, it also creates the RelRec that's needed because of the parent. And so it knows these types of what I consider to be specialized domains that normally requires extra programming. The other domains, there's a lot of macros, there's a lot of standardization that can be done, but RelRec and findings about usually is a little more specialized. So it's really happy to see that it has the capability and features for that. Continuing on, we have the control terminology. So this is the level after metadata mapping. And so this really shows you the granularity. And so as we know, everything everything, all the variables, they kind of fit in buckets. So we have conventions for the variable names, the types. We also have conventions for the control terminology. And so this enables you to have that level of viewing and comfort to make sure that the control terminology is actually in play. And this is done at a domain and the variable level. So you can see that you can expand on any of those domains and view it. And so after we're done with that, then the section on derivation, as we can see, these are typical things that will be needed across many of the domains. And so the system recognizes that. So this is where metadata programming comes into play. So the system knows which of the domains that have the date variables, and then it pulls in, because everything is standardized. So the domains that have the date variables, so they have a start date, end date, and then it knows those are the ones that need to have the study day. And then so other things that can be pulled in. So it's really nicely designed that way. When you go through this iterative process, what you have as the final outcome are the XBTs, SDTM, which is very easily downloadable. And so through this, you can go through each of the domains, you can kind of view them, and really serves as a nice cycle process, again, all within the system. The real charm and real uniqueness also with the system is that you can export metadata mapping specification. And so this is extremely useful. As you see here on the very bottom, those sheets represent the same steps that we just went over, metadata mapping, transposing. So basically you have a complete audit trail of everything that you just did through the user interface. Everything is all documented over here. So this itself can be read by SAS programs to convert the raw data to SDTMs. Again, this serves as documentation, and it's really ideal for traceability and compliance. So really like that, and that has that feature. Once the SDTMs are produced, we can go back to the study process flow and then take a look at the some of the details over here. This just shows you the directory where the UDM files are created. So as I mentioned, this is an intermediate step that's there and readily available. So that's the folder that has that. Within here, of course, you have the XPTs, SDTMs are readily available. And the reason for going back to this path location is really to take a look at the exporting, creation, and then fine tuning, for example, the define XML. And so within the system, we have this feature. So organizations don't have to write complex macros or go out of the system in order to create a define XML, and not just the basic define XML, but one that's in sync with the SDTMs, and it has all the hyperlinks, everything available within the system. It has the Excel file specification. So this is where you can make some edits, fine tune, make sure that everything is set up. Again, for that first study, the control terminology, any type of derivations that's there, you can see the granularity, the details of what's available in this package, because the final result, the define XML file, again, has to be go through compliance. And so the control here is really really superior, meaning that enables you to make sure that the define XML is in fact very comprehensive. The documentation, the linking, everything is very well documented. So this is a really nice, great feature that's there. As with anything, especially things that are produced automatically or by tool, compliance is paramount. So one of the things that I've always done going through things that have been produced from the tool is making sure that these principles are being enforced, namely traceability, as I mentioned. So even though programmatically we can actually see going through line by line programs and everything like that. Here, traceability is there with the documentation of the Excel file, because everything before that Excel file was done through the user interface, and that Excel file really assures us that we can review at a domain variable level the complete traceability. And that's very important. Reproducibility, again, the whole concept of leveraging metadata to reproduce. So as we have some changes or updates to the raw data, or maybe some updates to the raw data structure, we can see that actually translates down to the SDTM. And so we have complete reproducibility conformance. As we'll see in the next slide, you'll see that we can see the results of the rules, just like very typical we see in the industry for checking the conformance. So because there are hundreds of rules that need to be applied, completeness, all of the raw data needs to be standardized, and also we have integrity. Because of the early exploratory analysis, basically you have all types of users being able to see and view the data interactively. So that enables much more control of the higher quality that's there. Now, if we take a look at the compliance, so we see that Xbiom keeps in current with the rules that are there. So FDA, others, and then this is what the dashboard looks like. So here in this dashboard, you can see that it's a drill down feature, so it's very comprehensive. The nice thing about this, again, with a drill down, you can get down to the patient level, you can identify. In addition, you could also document what could be the underlying issue. And of course, any of the things that can be fixed can be fixed upstream, and then repeat the process to make sure that the issue has been addressed. The other nice thing I really like about this feature is that you can actually see a group of related issues to see how what's going on. So if there's a common theme that's going on, you can identify that. But again, this is all within the system, so you don't have to leave Xbiom. And I really like that, so I can actually see what's currently going on as far as compliance issues, and that's extremely important. With that, again, with the cycle process, you can fine tune and address those issues. And then next thing would be to download, export the SDRG, which would be typically the case. So very easy in order to do that. Then what you have is the actual SDRG. So this is the Word file. And again, you can see how metadata again is fully utilized. The only way that the system can generate this type of file is that it knows and recognizes all of the compliance issues that were there at a domain level, and documents that in here in the SDRG. And in addition to that, it will pull in any comments that were made within Xbiom system at the detail granular level. And so it aligns it. And so what happens is this becomes again in sync. So what you have is you have everything in sync with the compliance results and the SDRG. And so this gives you a way to dynamically and easily produce. So you can keep this in mind as a cycle where we go from beginning to end. That enables you to really shorten the cycle to reproduce this type of process. Now, many organizations may have may fall into maybe two types of categories over here. They may have no macros or no metadata file system, or they have minimum macros compared to alternative of having a full-fledged macro library or leveraging a metadata files or system. And so obviously then Xbiom works extremely well if you don't have or have minimum SAS macros and infrastructure for this. Many of the macros that are there in many of the organizations, they do minimal or basic level of metadata type processing, not necessarily the extent of what's being done with Xbiom. So actually Xbiom offers a really nice off-the-shelf GUI type interface to this metadata and the data with the exploratory analysis. And then with organizations that actually do have some macro library and things, as I mentioned, with Xbiom, there's actually best of both worlds in that there's areas where snippets of code can be applied in the pre and post processing in order to facilitate that. But you can see that the Xbiom offers a way to further enable programmers the ability to do that with the user interface and less with low code. As we get towards the end of this presentation, just going over a summary, hope you can see that there's many features in here that enable the benefit of standardization with the automation and compliance. The directors are better able to manage projects with automation and standards, as we saw. The cycle to create these submission deliverables is directly reduced from initially months to weeks, and then within the repeated cycle, the final version can be done within days easily. And so you have a very statistical computing environment, integrated tool that handles and can deliver all the submission deliverables in a reduced time and budget as well as resources. For the programming team, it offers a low code way to do this, so reduces the need for writing extensive programs, macros, faster SDTM, defined SDRG, as well as the unique feature of auto generation of SDTM mapping specification, which is extremely nice to see. For the scientist, what you have is early detection, early access to the data, which is paramount when you're talking about working with biomarker data, any safety type issues. That's because you have faster ingestion, curation, harmonization of the data. In addition, you have a great user interface to create SAP cohorts and really review that in conjunction with statisticians having the predefined templates, which really drastically reduces the time to tables, listings, and figures. As we take a look at it, there's some consistency and patterns as we take a look at specific domains, how we see the data. So why not leverage that with predefined templates that can be easily customized. And the nice thing about it is with the drill down facility from summary to the patient level details. So now I'd like to turn it back over to Rahul.
Thanks, Sunil. That was really well done. And I think in the last few minutes that we have here, some of you might be wondering, at what stage does Xbiom Mapper accommodate trial data? And the answer is whether you're in the middle of a trial or if you have legacy completed trial data, there are two things to consider. And many of you will have your own files of raw data in a file store with predefined cadence of receiving EDC and biomarker data and prescripted macros. And some of you are purely focused on the standardization of your data following a database lock and may have some complex biomarker data that is proving challenging to standardize. So no matter the data source, complexity, or stage of your trial data, the Xbiom Smart Transformation module can support your curation workflow, and we welcome the opportunity to walk through that with you. Just email us at ask@pointcross.com, which you'll see in the next closing slide. We have received several questions throughout today's discussion, and because we have just a few more minutes left in the conversation today, we thought we would address a couple of them. Number one, we received a question asking whether Xbiom uses SaaS under the hood and whether it means that you have to have an expensive SaaS license. And actually, no, on the contrary, Xbiom does not use nor rely on SaaS for generating SDTM. Xbiom itself is built on a big data stack, and under the hood, it uses Hadoop components such as Spark and some other controls for advanced users to write in PySpark, Python, and R. So we hope that that answers that question. We'll also be providing written responses, as we mentioned, along with links to the upcoming webinars that we have scheduled and lined up, as well as download links to the 2 PDF flyers that you have referenced here on this screen. We're asked whether Xbiom is also an MDR, a metadata repository. Xbiom, yes, does include the complete metadata repository capability, as well as several controls for users to perform impact analysis, differences of changes till the attribute levels, etc. And the export of mappings to Excel is also really useful for documentation and traceability. So I think if we have any other further questions that come through in the next couple of minutes, we're happy to answer them. And you can, again, use the Q&A chat function below. We encourage that to continue to flow in so we can collect them, anonymize them, and provide written responses in full and in detail. So Sunil, is there any other closing comment that you'd like to offer before we close?
Yeah, thanks, Raul. So again, I appreciate your time and interest in this topic. I'm really happy to see that Fuse recognizes as a hot topic. I'm very anxious to give the Wednesday webinar next week on this, so please join me again there. You see the links to the process flowcharts. I hope that this has been very informative and eye-opener to see the possibilities that's there as far as a future state where metadata-driven process is actually possible with the controls and the compliance in place. So I'm really excited to share this information with you, and really appreciate that Point Cross asking me to provide this information. So thank you.
Thank you, Sunil. And I want to thank all of our participants from around the world that have joined us today. We'll make a recording of today's session available, and we'll send a follow-up note to all of you who have registered to participate with the link to that recording as well as the written questions. So again, allow us to thank you. If any other questions come up, or if you're thinking about today's discussion later on, please just drop us an email. Again, ask@pointcross.com. That'll go straight to us. You're also welcome to reach out to Sunil directly at the lower left email address that's been provided. So again, I want to thank everyone and have a wonderful rest of your day and a happy new year. Thank you again, Sunil.
Thank you. Bye-bye.